Hello again. Due to timing limitations with Screencast-O-Matic, I am going to be presenting the second half of my honors thesis presentation right now. So continuing with the results. My study found that bluegill were able to improve their learning score the following day. I mean the second day of learning. So this graph shows the number of escapes for day 1 of the learning trials compared to day 2 of the learning trials. This data shows that there was a greater number of escapes on the second day of the learning trials, indicating that bluegill were able to improve their learning scores after the first day. So this was very exciting because it really did show that bluegill are able to learn a novel task. To begin with my discussion, the results of this study raised the question of the possible reasons why the only successful learners were the less aggressive individuals from the aggression trials. One reason for these unique results is the phenomena of behavioral syndromes. Behavioral syndromes are suites of correlated behaviors that account for differences in personality, and these are analogous to human personality traits. This accounts for the reason why individuals of the same species can act differently to different situations, just as humans do. A commonly researched behavioral syndrome is the shy-bold continuum. Some individuals or genotypes in a population are generally more aggressive, active, exploratory, or bold, while others are generally less so. And several studies show that bolder fish are generally more exploratory than timid fish, and are also more aggressive than timid fish. My contradictory results from my study compared to previous studies could have been due to the laboratory setting. An experiment with pumpkin seed sunfish, a close relative of bluegill and bluegill sunfish, found that differences in individuals concerning the shy-bold continuum are stable in nature, but disappear in isolation in the laboratory setting, which could account for the less aggressive fish being the only successful learners in my study. There were also more escapes on day 2 of learning compared to day 1, showing that bluegill in this experiment displayed learning capabilities when given a task. Other tasks that have been learned by fish that were not previously mentioned are maze solving and conditioned avoidance with light. And this conditioned avoidance with light was very similar to how my bluegill learned to associate the presence of light with needing to swim through the divider to escape possible chasing and stress. In addition, a greater number of escapes on the second day of learning, indicating less failures, shows that bluegill have memory abilities, although the memory window can vary from species to species. Lastly, a third day of the learning trials was not necessary, as a previous study showed that the results of a novel learning task were nearly the same on day 3 as they were on day 2. So this is the reason that I did not continue the learning trials any further than 2 days. To continue the discussion, I tested many variables in this study, but several were not statistically significant. There was no significant difference in learning ability or aggression scores based upon which experiment was conducted initially, either the aggression or the learning trials. Certain behaviors, such as aggression, are known to be affected by a prior contest experience, so it was thought that individuals who lost in aggression trials would perform poorly in the learning trials, and that the winners would perform better in comparison. Similarly, it was thought that individuals who performed well in the learning trials first would be the winners in the aggression trials, and that the poor learners would become the losers. In a study with mangrove rivulus, it showed that individuals who had recently received a winning experience were bolder and quicker to accomplish a learning task, while the individuals who received a losing experience behaved the opposite. In addition, another study with fruit flies showed that as hierarchical relationships were set, winners attacked more and retreated less, while the losers attacked less and retreated more. Neither of these phenomena were observed in my study, and the statistical tests were not significant. Another variable that I studied for was not significant was the differences in relative weight between pairs in the aggression trials. There are correlations between body mass, standard length, and boldness score, suggesting that fish with good body conditions are bolder and more exploratory than fish with poor body conditions, even though this phenomena was not observed in my study. Several studies have also shown that fish with larger body sizes will be more aggressive during a fight or confrontation for territory and will outcompete their opponent in most cases. And this is especially true for bluegill and bluegill sunfish. One reason for the lack of significant results concerning body conditions of bluegill in my study could have been due to stress and prolonged holding time in the laboratory. Stress correlated with holding a fish can have negative effects on overall health, such as suppressed immune systems, decreased growth, decreased swimming performance, and even death. So this additional stress due to laboratory holding environment could have likely prevented my bluegill from adapting and therefore prevented them from exhibiting normal aggressive behaviors associated with body condition that would have otherwise been observed in the wild. So now for the conclusions that I gained from my thesis. This study successfully showed that bluegill are capable of learning a novel task and can improve their learning score the following day. So my thesis really showed that even non-mammal organisms such as bluegill are capable of learning a novel task when presented with one. And this is really important because this can show that bluegill are able to adjust their behaviors, to adjust to changing situations in their environment, which would hopefully in turn mean that they could continue changing and adapting to changes in their native environments, leading to longer, long-term survival. I wanted to take the time before I end this presentation to give thanks and acknowledgments to people who made this possible. I first want to start with thanking the committee who granted me the Student Engagement Research Grant. This was a huge part of my thesis because it gave me enough funding to buy all of the necessary supplies such as tanks, dividers, and filters in order to make this whole thesis really come to life. And next, I want to thank faculty members who helped my thesis come along as well. Dr. Angela Lamora allowed me to take over her anatomy lab for the summer to house all of my fish and equipment. Mr. Paul Liga was helpful in aiding me to gather equipment and supplies, along with making sure the lab and my fish remained undisturbed throughout the data collection process. Dr. Michelle Schutzenhofer has been my academic advisor since I was a freshman here at McKendree, and I am so thankful for her guidance and wisdom in not only succeeding academically, but being able to reach my career aspirations. I will also always be grateful for the times that I was able to go into the field with her to learn more about ecology and the beauty and importance of bees. Lastly, I owe a huge thank you to my thesis advisor, Dr. Michael Lewison. After taking his first animal physiology class in McKendree, I was excited to have the opportunity to work with him and learn all about the research process. I may not have been the biggest fan of working hands-on with fish at first, but I learned so much and was able to create this thesis presentation to share with the McKendree community, and I could not be happier with how it all turned out. His unending support and knowledge as a seasoned research veteran was a huge part of my honors thesis, and I am humbled that I got to be one of his first thesis students at McKendree to blaze the trail for future students to do the same. Thank you all so much for tuning in to my presentation.